thanks, Gordon, for the invitation. As I said to you, and you already repeated, I really feel it's a fantastic opportunity to talk about translation research in this place. And Glenn, you've been more than generous in your introduction. So, few comments. First of all, you can say that I'm undisciplined. Obviously, right? I'm working on so many things that are not really making a single story. Or you can say it's multidisciplinary. And then you immediately need to say the next thing, that there is no way I keep all these things in my head. You can't be experts in so many things. So it's another way to say that when you do multidisciplinary work, you do a teamwork with many people who are equally important for this work. So that's actually a very good way to start what I want to talk to you about. I have to say something about winter. So I told some of you, this is my wool jacket. First time I wore it this winter. I was particularly excited. I saw a small dirty pile of snow on the side of the road, the only pile of snow I saw this winter. I also don't like snow, I have to say that. And I appreciated in particular the visit to Dr. King's lab, where I walked to a room that is just the right temperature, 30 degrees Celsius. So that's, so all this stood in the way of my visit here. And truly, I really think that it's important to come here for the obvious reason. How many of you took a class in physiology? Ever? How many of you took a class in pharmacology? You see, if I ask this question anywhere else, I'll have the physicians that will be hesitant to admit, and that's about it. And right there, it's a major reason why this place is so important, and how you, the students, are skilled in things that are no longer taught anywhere else. We don't teach physiology or pharmacology to our students. Medical students get it. If I try to force a student to take any of these classes, it's really a big fight. And importantly, it means that the kind of research that we do in academia is so skewed because we look at our molecule of interest or cell of interest and think about the animal as something that holds this molecule, this cell, and nothing beyond that. We kind of forget about what it is that we're trying to do, especially if we're thinking about anything that will have eventually some utilities, either in animal care or in human. But more importantly, even if we do basic research, it has to be in the context of certain reality. And the reality is the whole organism. And as I said, when you are applying for jobs, for postdocs, or for jobs anywhere else, you emphasize this point. You don't explain it away, which I see sometimes students do, but you actually emphasize that not only you bring your skills as molecular biologist, cell biologist, or whatever you've done, or chromosome karyotyping something, but you actually also know about physiology and whole animal anatomy and so on. Those are skills that are extremely valuable, so don't hide them. This is you have something special here. I also think that in this school you have another thing, and that is that you have a disease in animals that occurred naturally rather than somebody induced in the animal by giving them a tumor and so on. So that's another thing that is a true asset, and those are the reasons why I think that if you guys decide on translational research, it's going to be a fantastic opportunity. All right, so I have a lot to talk to you about, and what I'm going to do is I'm going to, unfortunately, not give you a true science talk, which I prefer to do, but I will tell you about an old science talk, old science discovery and how it translated to clinical trial and to the startups that I started unintentionally. And I will tell you then one more story, and then finally I'll go to Spark. So let's start with this little slide that reminds me to tell you about something that in the US lots of people know about. Okay, I'll read my slides here. But people are talking about the value of death, that we academicians keep discovering cures for everything, but actually industry is not doing it. And Zarkuni was the NIH head, has made a lot of publication and write-up about that. Now he's, by the way, in Sanofi, so hopefully he's going to make true with his promises. But the bottom line is that we are faced with a true challenge. We're talking about two cultures that don't have much in common. So in academia, We are really, we have a particular scientific questions that we started to study either because accidentally we started our PhD or our master degree because we liked a particular professor, or because we really have a passion for it. And we followed that question over the years, and sometimes we get distracted, we go to another direction because we got something else that was interesting to us, but it's really kind of a random walk. And importantly, the way we are rewarded, the way we get tenure somewhere, or the way we are promoted in academia is based on the fact that we've done something nobody else has done, that it's a true breakthrough and it didn't follow anybody else's. That's how we are rewarded. Then you have the industry that has exactly the opposite culture, where you are starting with what is it that you're going to develop? How does it look like? Is it going to be a pill, or is it going to be a syrup? Is it going to be given twice a week, or it's going to be given every day? Is a clinic in Africa, so it has to be, you know, resistant to 36 degrees Celsius and humidity, or is a clinic in Canada, and so on and so forth. You start with a product, and then you walk backward. What is it that I need to know about this product in order for me to put the gazillion $1 million to develop it? Very different from us, where we started with, oh, I'm curious about this. Oh, now I'm curious about something else. Let me spend some time on this, and so on. Okay? So this is different. And once you prepare yourself a path, because you walk backward and you figure out the way how you go eventually to your end product, you created a path with a lot of decision point where you're going to drop the project. This path is predetermined, and you have to agree with many experts in the fields about that path. So again, random walk, not acceptable. And finally, because the amount of money that you invest in developing a project is so large, and your chances of success is so low, and I quoted a number of numbers, but let's say 1 in 100 projects in preclinical eventually get to approved drug. I think this number is incorrect. It's more like 1 in 1,000. But whatever the number is, it's still your chances to fail exceeding by far the chances to succeed. So by nature, you have to be risk averse. So think about those two cultures. And we're trying to do translational research. We're trying to take academic work culture and bring it to industry. So huge gap. And the only way to start to close this gap is to understand that, first of all, we don't talk the same language. So when we're saying translational, we're really talking also about language barrier. Okay, so the 3 stories I'm going to cover is the first one on how we started to develop a drug for myocardial infarction. And the second one is just which eventually led to starting a company and so on, and I'll tell you about that. And then I'll tell you a small nugget about something that has nothing to do with that, just to show you that you don't need to be an expert in any field in medicine or any field whatsoever. You just need to know who to ask in order to do something else. So I'll tell you about the leishmaniasis project that started from our research, and you'll see that it is logical. Even though it seems illogical, there is actually a very clear path and typical for industry. And finally, I'll tell you more about Spark. So let's start with the first one, basic research in the lab, and eventually how we develop a drug. So briefly, and hopefully you've heard Glenn talk about these topics before, so you know there are many protein kinase C isozymes. I started to work on it in Dan Koshan lab in '83. We didn't know that there were many enzymes, but about '88 we found out that there are multiple enzymes, and then shortly after we started to discover that the enzymes in the dish look like, or in the test tube look like the same thing. They do the same things, they are activated the same way. It's very confusing why we have multiple enzymes in different tissues. And when they've been cloned, it was clear also that they are very similar, not only in their regulatory end, but also in their business end, especially the catalytic side. And lots of companies were interested in figuring out what makes them different, and are they individually involved in different diseases. I wasn't interested in that. I was just interested in basic research question. Why do we have many enzymes? What makes them different? And one of the things that I've done, even though I'm a protein chemist in nature, I was actually looking. So here is a grant. I think it's my very first grant from the NIH on protein kinase C. From '89, and like you PIs here, the last line is always about, okay, and if I figure it out, there will be a cure. Right? That's what we do. We take the public money, And we promise them we're going to deliver something. So I want to make one point, and if I got only this point across, this is a one, which is you in Canada, we in the US, are counting on the public to fund our fun. We're doing basic research, we're teaching. We are really enjoying the public trust that we're going to do good with their money. And the good thing to do with it, the social responsible thing to do, not as an individual, but as an institution, is to take discoveries that we've made and to do our darnest to make sure that if they have utility, that this utility will come forward. So I don't say that individuals need to do translational research. And in fact, I would argue that there are many people who shouldn't be doing that. That's really not their talent. They are not working in large teams. They really like to work individually. That's fine. But we as an institution need to put an effort in taking this discovery and bring them back either to veterinary care or to human care because we got the money from the public with a promise that we were going to do something with it like that. We didn't get the money for the sake of acquiring knowledge. Nobody writes in the last line of their grant proposal, and I will know more after these studies, right? Okay, so now to what I saw. So this is a study from '88. We looked at variety of cells in culture. We generated our own antibodies at that time to protein kinase C. Those are heart cells. They are primary cells. Everybody was then working on tumor cells. And I was lucky enough to work on real cells, so neurons and heart cells and so on. And what I saw is this. I had monoclonal antibodies to 3 enzymes here. This is before any treatment. This is after treatment. And excuse the fact that you can't see much. You know, this is really an antique now, probably older than some of you guys in the audience. And I'll just show you one more picture here. That first of all, it shows you that before and after stimulation, in this case with hormone that activates protein kinase, the alpha-1 adrenergic receptor coupled to GI, showing that the individual enzymes start from one location and end up in another location, for example, internuclear mainly to perinuclear and fibril structure. I also want to show you this guy and to tell you how clueless I was about what I was doing. I was using heart cells in culture, and I saw this kind of staining, and here is a better staining. I saw this cross striation. I'm working with heart cells in culture. I never took physiology in my life. And I said, what are these bands? Okay, I would say that I'm not rare. And this is another example of what I was trying to tell you. This is terrible. I was then a professor at UCSF, and I asked, what are these bands? I bet most of you would not ask this question. So this is another example of why what you are trained in is really unique. Anyhow, so the bottom line is individual isozymes, when you look in the microscope, before and after activation are localized to different sites. And importantly, at that time, we knew that protein kinase C is activated by binding to lipids, because the second messenger that activates is lipid-derived messenger. And so people didn't see it because they didn't use cells in culture that are real cells. They use cancer cells that divide quickly and don't have time to create all sorts of organizational structure in the cells. So we propose that maybe the way the enzymes work differently is that they are localized to different parts of the cell. This one on cross-striation, cytoskeletal element. This is this fibril structure. Now we know mainly mitochondria. And this is on the plasma membrane and the perinuclear membrane. They see different substrates. They do different things because they are localized to different parts of the cells. So that was the basis for what we call the RAC hypothesis. The RAC stands for receptor for activated C kinase, which was the name of protein kinase C in the 80s. And the idea is simple. Each enzyme has its own set of RAC in different parts of the cells. When the enzyme is activated, it binds its RAC and not any other RAC. Basic research concept. And in order to prove the hypothesis, which, by the way, was really against the dogma, you need to show that that's the case. In other words, you need to show that if you inhibit the localization of, let's say, the yellow isozyme, you lose the functions that related to nuclear function, but not cytoskeletal element, whatever, contractility or functions related to ion channels. And importantly, if you can do it, you should be able to activate just one enzyme and show that if you now tether it to the right place, it carries out those functions, and the other enzymes do not. So there were really tools to answer this question, okay? Not really any drugs. And here's the way we thought about it. I'm a protein chemist. And I thought that a lot of information will be in primary sequence, even though I came from the Weizmann Institute where everything was supposed to be three-dimensional. I started to think maybe not. So we developed a rational approach to identify short sequences that are critical for the functions. I'll show you later on one example. I just don't have time to talk about it. In fact, it's my favorite topic, but unfortunately, I don't have time to talk about it. So the idea is that you can have short peptides that represent the interaction site between the two proteins that are required for the anchoring, therefore do not allow the enzyme to come close enough to the substrate, and so the physiological response will not occur because you didn't phosphorylate it. And those short peptides can be derived from the enzyme or from the rack. Both will work. And you can also make activators. Since I'm not going to talk about activators, and since they are a bit more complicated, I will leave it there. But they are working with the same principle. They inhibit, in this case, intramolecular protein-protein interaction instead of intermolecular interaction. OK. So we made isozyme selective inhibitors and activators for each of the protein kinase C isozymes. And we started to use them. And we also started to give them to whoever wanted to give them. And it occurred to me that the only way that people start to value them is that we can see certain loss of function or gain of function from these peptides, not only phosphorylation. So I thought, OK, heart cells in the culture, they beat. Protein chemists can count, so we can see they beat fast, they beat slow, and that will be the function that we will follow. So we had a study in which we showed that delta protein kinase C and epsilon protein kinase C, both activated by the alpha-1 adrenergic receptor, have an opposing effect on contractile rate. So in the dish, one will slow down contraction and the other one will hasten contraction. And we can show it with the peptide inhibitors and activators. I said, wow, we can regulate heart contraction rate. So I put an abstract for the American Heart Association. I got it to give a talk. It was one of those 3,000 people room. And I was talking about contractility, but I actually meant contraction rate. I didn't know that contractility, even though it's a word in English, doesn't mean contraction rate, but actually mean contraction strength. That was my first mistake. And then when people started to figure out that I'm talking about rate, they were even less interested in kind of leaning back and, no question. It was just terrible. But luckily for me, a cardiologist that sat relatively in the front from UCSF, I was then at Stanford already, came to me and he said, Daria, we cardiologists really don't care about contraction rate. And I thought, well, how can you say that? I mean, I'm standing here, my heart is pounding, surely important, If I can regulate the contraction rate, there must be a clinical indication for that. And then he said, no, we are not really interested in it. We can figure out how to deal with that one differently. So I said, so what are you interested in? He said, well, we are interested in heart attack and how the heart is dealing with a ischemic event. I don't know how to do it, I said. He said, don't worry, I'll send you a cardiologist. And he sent me a fellow, Mary Gray, who was the first in then forever, at least one MD or DVM person in my lab. And she was the one who started to build the cardiac ischemia model. So now there is a relevant clinical use. So that's the first example of you can do something that seems intuitively to you to make sense, but you better talk to somebody who knows something about it and will tell you whether your intuition is correct. That was a case where absolutely not correct intuition. So from then on, I always talk to clinicians, and I told one of you that, for example, I walk to work and back, and I oftentimes manage to convince somebody to walk with me. Usually they need something from me, so I said, OK, you can walk with me. And the deal is half of the walk is you tell me something about medicine that you know. And some of them will repeat the walks. Others will be the last walk, usually because they complain that I walk too fast. But there you go. I think clinician DVMs know what the problem. And you have to start with a problem if you want to do something that is translational, but also if you want to do something that is important. So I could work until I die on contraction rate, but if it's not important, what am I going to say? Oh, I'm really proud. I figure out how cells beat in culture. OK, big deal. OK, so we started to work on cardiac ischemia and reperfusion. And here, quickly, way too slow. I'm going way too slow. You know what? Let's skip all that stuff. Trust me that we figure out that turning on delta protein kinase C at the end of heart attack makes a difference. So I'll give you just one example here. Here is a Langendorf preparation. And here is, you don't need to know anything about echo, or in this case, it's not even an echo, it's just a LV-gram. And you see that this is really green, and this is really healthy, and then in heart attack, in this model ex vivo, nothing, and then it starts to pick up, and this green is really poor, this blue is really poor, okay? So you can start just with looking at the color. And here is the same model where we put delta protein kinase C inhibitor, so downward red is meaning inhibitor, right when we started to put, again, oxygen and nutrient in this ischemic heart. And you see that this green is now way bigger than this guy and start to look more like normal, okay? Then I don't need to be a cardiologist or be able to read these figures to say something good is happening here, okay? So this was in animals, in rats and mice. We also gave this reagent to anybody that was interested. I went to the American Heart Association, showed this data, and they say, no, Daria, reperfusion injury doesn't exist. Hearts die from ischemia. It's because you did it in ex vivo, and this heart is not working, and you have to do it in vivo. And by the way, rat is not the right model. You need to do it in mouse, in rabbit. So everybody that would say any of that, they would get in the mail 1, 2, 3, or samples labeled 1, 2, 3, and say, okay, you do it in your model, and tell me if it works or not. And at the same time, so you get other people to actually do the work for you. And importantly, especially if it's somebody that doesn't believe in it, then you feel more confident, because I have to say that I always have a problem that before I publish a paper, I'm convinced of the data. Once the paper is out, I start to say, is it really like that? So every time somebody else does it and finds the same thing, it's really a relief. Okay, so we, of course, we're a basic research lab, so we looked at the molecular basis and cellular basis, and that's how you publish work on it, and it was really fun, but it's not really translational research. So we have to talk about people who will be interested in translating it, and the first thing they say, mouse, rats, not relevant. You need rabbit, and you need, I don't know, guinea pigs. So I said, oh, we have it for so-and-so published in rabbit. No, you need larger animals. So you guys call larger animals horses. Well, luckily, they considered pigs large enough. Is that large animal? Okay. Why pigs? Because Stanford has pigs in the basement under the angioplasty lab because they develop angioplasty devices. And by the way, I used to use this pig picture since we did this work. And I used to say we used 25 to 40 kilo female pigs. And finally, somebody gentle told me, you know, this is not a female. Okay. So how did we cause heart attack in this animal? We thought that it would be rather important to cause a ischemic event, the stop of the blood flow. I assume that you all know about heart attack, so I'm not going there. But stop the blood flow from within. So we use the same balloon catheter that is used to open up occlusion, and inflated it for a duration that will give us infarct size in the pigs that is equivalent to what you see in about 4 hours in humans. It's actually relatively brief. It's about 35 minutes or so. Okay, so now remember, I want to give the drug at reperfusion, but now I have to do it in pigs, and I made the drugs, the peptides, in my lab. It cost me money to synthesize these peptides. So I didn't want to give the drug systemically to the pig because this is a 30, 40 kilo animal. So I thought, can we deliver it directly to the heart? And I looked at this device and I said, oh, look, I have here a particularly useful lumen. This metal piece, if we pull it out, we can deliver things while the balloon is inflated. So that's what we did. We pulled the guide wire out, and we delivered to the pig, to the post-occluded area. So I delivered directly to the area that was ischemic, and I delivered it while there was no flow. So I could leave the balloon inflated and deliver the drug to the post-occluded area and then see if it's beneficial. Okay, so now come the next step of translational research. The first experiments that we decided to do with these pigs was to hold the balloon for 10 minutes. So deliver it at 25 minutes and leave it on for 35 minutes and see what's the benefit. So the guy who was doing it was an interventional cardiologist trained in Japan, and he said, 10 minutes, too long. Okay, so started to negotiate. Is 5 minutes okay? 5 minutes, too long. So, and that's the negotiation continues, and finally I said to him, so what would be short enough? He said 2 minutes. I said, okay, how about we'll do 1 minute? Okay, so we went for 1 minute, and the drug was delivered for 1 minute at the end of the occlusion period, and then we measured cardiac function and also infarct size. You can slice the heart and measure infarct size by doing stain. Of course, in patients, you would use other methods, including MR, but that's what we did in the pig. Okay, so first let's look at infarct size. This is looking at 5 days. And in this case, I think, yeah, it says we had a dozen animal in each group. The reason why we have large numbers is not because we didn't see a difference in 5 animals. So if I was just thinking about publishing a paper, I probably would stop at 5, considering that this is a $1,000 animal study. Anybody should stop at 5, but we did many more things with these animals, so we kept adding animals. And luckily, actually, that was the year that I got the endowed chair, so the endowed chair went to the pig. I always say that that was a good use of the money. So here is a infarct size in naive animals, and if they were treated for 1 minute with this drug, the infarct size went down by about 70%. And here is what when we're looking at ejection fraction, which, as you know, is a reflection of how good the heart is pushing blood through each heartbeat. You see that animals like human, actually, the pigs start with about ejection fraction of about 70%, 65%. And here you see 40 animals or so, and we followed them post-infarction. And in this case, we stopped after 14 days. And the reason why there are fewer animals in each stage is that we harvest them, or whatever the word you use here, euthanize them. How about that? And to measure infarct size, so that's part of the reason. But also notice the real science here. Here are not inbred pigs, and you see that some of them start with ejection fraction is much greater than others. The bottom line is, if you do statistics, you see that there is a very huge decline immediately after infarction. This is a few minutes after infarction. And then 1 day later, there is a tiny improvement. By 5 days, they get to about 50% or so ejection fraction, and they stay like that. If they were human, they would be sent home with beta blocker and statins and bunch of other drugs, ACE inhibitors, because they are candidate to heart failure. So what happens with the animals that got 1 unit of delta PKC inhibitor? Well, they start as spread as a control, of course, because they are just, I chose one to one and one to the other. And then immediately after infarction, they already are as good as the animals without the drug or with actually controlled drug will be. This study was done in a blinded fashion. The first 5 animals, the person who did the angiogram was doing also the echo, and they could see right away which animal was treated or not by the response of the animal immediately. And so we started to do the following thing. They will do the injection. If there was only one interventionist in the room, we will cover it with a paper. So they have to work the rest blinded, because otherwise they will be unblinded, just looking at the data. But doing blinded study is really critical. And if you can do it from the beginning. Even though it's frustrating, your graduate students to no end, do it. Okay, so the bottom line is by 5 days, they looked like they didn't have heart attack. And they remained like that. And we actually had few studies that lasted longer. So I started to think that this is really working. And any fool from industry would see that there is value in it. Right? We wrote patent about it, and then the Office of Technology Licensing started to talk to people about it, and nada. So obviously I thought, Office of Technology Licensing is not doing the work. I'm going to do it. So I went to talk to everybody I knew and many people that I didn't know from pharma, and they told me that they were not interested. Okay, I'm going to skip that. So the reason why I had to talk to pharma is that I recognize that there is no way we can do the translation research that is required. It requires many things that I don't know how to do. And importantly, in academia, you traditionally don't do that. And the cost of that is so large that basically we kind of expect pharma to pick up these findings, this early finding, and do the rest of the work. So when I visited the industry, I got a lot of specific responses for why they were not interested. And I'm going to give you this one because I think that this is really an important, it was an important lesson for me at least. So first of all, they said that they don't want to work on protein kinase C inhibitors. They spent by that time 15 years in making protein kinase C inhibitors that works on the catalytic site and killed many animals. And with one exception of lilies that maybe have a compound that maybe is selective, that we all know is not selective, but so be it, for the catalytic site. And then they said that they are not interested in cardiac protection for ischemia and perfusion because here is a president of the International Society of Heart Research writing a paper that says that protein kinase C has the same number of papers that say that it's bad or good or not at all affecting cardiac ischemia and perfusion, so it's a wrong target. Obviously, otherwise the literature was not so confusing. However, they did work with non-selective inhibitors, not isozyme selective inhibitors. And then I'm working with a peptide. Peptide is not a drug by pharma, at least definitely not at that time. And then it's a peptide that works inside the cell. Peptides do not cross biological membranes, so it can't be working. And here, information doesn't matter. So the fact that they saw the data, it didn't matter. They know better. Those things don't work. And then I made a protein-protein interaction inhibitor. At that time, there were no attempts at that because it was so considered very flat surface and you can't make drug to it. So basically, they say, go away. OK, so I came back to the lab and I said, OK, guys, not going to happen. And I had a graduate student, Leon Chen, in the lab. And he was the one that actually organized the work around the PIC team. And he kept saying, we have to start a company. We have to start a company. I said, I'm not starting a company. I don't want it. I'm an academician. I don't know anything about it. And he kept asking, saying, and so on. Finally, I said, you know what? When you graduate, I'll do it. I'll try to help raising money. Thinking, you know, I have at least 2 more years. You know, some pharma will eventually take it. Big mistake. So this was this conversation in October. In November, he went to his committee and said that he wants to graduate soon. Can he have a committee meeting? He had one in December. January, he graduated. And I told the committee, hey, you know, he's not ready. He was ready, and we started CHI. So this is CHI. CHI stands, by the way, to kinase activator and inhibitors. And the way we started CHI is, first of all, go and talk to a gazillion million people. I talked to anybody, including quite a few people at Stanford who were entrepreneurs themselves. And they all told me, don't do that. It's too hard, or it's too simple, or it's not going to reward you because you're an academician and it's not intellectual, or everybody is out there to get you and steal your money and your brain, and all sorts of unhelpful comments. The most important conversation I had, actually, so I really talked literally to everyone. And the most important conversation I had is that when I took out the garbage at home, which I usually don't do, and I was pulling out the big garbage can out for the collection, and my neighbor across the street was doing the same. Hi, Ken, how are you? I'm fine. How are you? Fine. What are you working on? Anyhow, Ken was in devices, business development guy, and I said, you know, I have this problem. And now Ken was our first BD person. He was the one who got us corporate lawyers to get the company started and so on. So next to the garbage can, CHI began. And the thing is that when I was asked by investors and anybody else about why I'm doing it, it was really both Leon and I, who was employee #1 for a long time, we're focusing on, really, we need to get it to the patients. I have a father who died of MI, I have a brother who had an MI, I know of colleagues that have MI. This is worthwhile. My 2 Japanese fellows, cardiologists, the interventional and the general cardiologist, both told me that they will take the drug first before they go to the ER if they have chest pain. So, you know, and this is high bar, really high bar. Okay, so that was the motivation. So we went to raise money, and I have to say that we went to raise money is, how about you start with having absolutely no ego? Because you go to the room, you waited for this meeting for a long time, you prepared your darnest, and people are sitting around the table. Some of them are asleep or yawn. Some text while you're talking. Many leave the room and come back. And then they ask you something and say, oh, if you don't have an answer for that, we're really not interested. And then the next one will ask this, you have an answer for that. They don't care about this answer, they want another question. So it's a lot about, even those that were sitting in the room and were attentive, it's what is in their head. You don't know what it is, what they are looking for. And it's a lot about not you. So it's not that you became stupid there when they turn you down. It's just that they are not interested in what you're telling them right now. On their mind is another application, another drug, another company they're working on. They don't have much money, whatever. Or they were just irritated by their spouse in the morning. It's not about you. So you have to go in knowing that you will hear more no's than yes. And I have to say, at that point, I was 10 years at Stanford, didn't used to get too many no's. So if I had a fragile ego and I would start to second guess whether it's good or not, it wouldn't have happened. But I was just, you tell me why not. And why not was very helpful. A lot of people actually gave me good advice. Boy, I will never finish this talk. Anyhow, so this is me activating the clinical trial site, the first site, in September of 2004. I took a year in the company, learned a great deal in the company, and then came back after a year to Stanford. I'm going to skip a lot because that's the part that is about translational. The bottom line is that they were clinical trial in patients that looked good, then Big Pharma got in, and instead of giving the drug the same way that we gave it in the pig, which was intracoronary, they decided to go IV, and then they end up treating 1,000 patients, which is a lot, with 100-fold more drugs than we use, which is a lot, showing absolutely no toxicity, but unfortunately also no efficacy in this large patient population, statistical. But 10% of the patients didn't have MI. They are still included in the statistics. 10% of the patients didn't have reperfusion because the occlusion was permanent. And the majority of the patients have tiny, tiny, tiny infarction. So it's difficult to show benefit on a tiny infarction. So this is another way to say you can put your heart in on something and that eventually what happens is not in your control. So that's another thing about it's not about you. So what am I doing now? Amgen bought the company for another program, which is another good story about basic research going translational. And Amgen now, I'm trying to push them to go back to these assets because they have all these assets and they are considering it. So hopefully something good will come with it and other PKC inhibitors. So okay, I'm going to skip all that. So all these people are many people that contributed to the work, and importantly, I'm showing you in blue the MDs or DVM. Quite a few people. Okay. I think I'll skip the second story. It's just the point of the second story is that you don't need to know much about a particular field, but you need to know who knows. And then you need to bring something to that field that somebody else doesn't know. So we use this idea that REC are important in Leishmaniasis because Leishmaniasis, I read, in reading about REC, I saw that Leishmaniasis has a REC. It's called LEC. Luckily for us, they call it the same name, so that's why I found it. And they have 4 copies of LEC, but if they lose 2 copies, it's dead. And it's in the promastigote and the amastigote. So I thought, okay, REC is important in Leishmaniasis. I'll make peptides that will inhibit the REC interaction with its proteins. But I didn't know which proteins they interact with. There was no literature on that. So I thought, okay, I don't need to know which proteins. I know how to design peptides, even if I don't know what's the target. So we made peptides, and we now have a large study with collaborators in Canada and in Mexico and in Brazil. And it's all because you know who knows what, and you go and ask them. So that was, in a nutshell, that story. The bottom line is that you can't do it alone. There is no way you can do it alone. And why do you get the attention of these people? Because first you call them up, and you basically have difficulty pronouncing the word, I don't know, promastigote, and they're kind of hesitant to talk to you about it. And then you tell them what you want to do and why, and then you get them interested in what you're doing. And then you expect to hear quite a few no's, which we did in the beginning. And finally, somebody says, oh, you know what? I'm going to try it. So this project that is actually starting in Canada, both of them in McGill, are actually they are going to use a large amount of time in their facility to actually screen some of our compounds. Okay, so now let's go back to Spark and why did I do Spark. So I was, when I left for CHI, for this 1 year in CHI, I was then the chair of the department. So at Stanford, the rules are you can start a company, you can own stuff, and so on. You cannot work in the lab on things that is going to be for profit. So all you do, it has to be basic research. If you have conflict of interest, you declare it. You saw my slide. And if you go to your own companies, you can't take sabbatical. You have to leave Stanford for that period. So I left Stanford. I was on leave for a year. And when I came back, I thought that, first of all, I had a blast. it was an amazing thing. I thought I have a lab that is a team lab, you know, that people are really working with each other, that they are actually good contributors to the overall product. I thought, you know, I really know what team science is. Not the case. So when you work in a company, you understand what teamwork means, because everybody's livelihood depends on anybody in the group. In fact, the dishwasher, the animal cage washer was critical in preventing a huge infection that the neighboring animal washer didn't prevent. So if we had an infection in our animal facility, Kai would have failed, as an example. Every single person is important there. And the other things that I learned is how incorrect the advisors were when they told me about why not start a company. So they say it's not intellectual, it's one foot in front of the other. Not true. They say it's intuitive. Absolutely not intuitive. They said it's, you know, everybody's out there to get you. No, you know, the VCs like us, they don't own this money. They took somebody else's money, convinced them that they're going to make them money by using this money. They have to pay this money back or to show that something shows for it, or they won't be VCs anymore. So, you know, it's really about trying to take an idea, developing it, and it is really worthwhile. And as I said earlier, I really think it's also our social responsibility when we can. So I went back to the dean and suggested that we'll start a program on that. And that was a mistake, because he also asked me to be a dean of research, and we discussed a little bit my negotiation skill of this dean of research position, which I think were better than yours, because I'm just dean for a day on Wednesdays, although sometimes it happens on Thursday, Friday, and Sunday, and Monday too. And the program, the idea was that there must be many more inventions like ours that are not licensing because they are deemed too early. And can we work on it for a while in academia to mature them so that industry can see its value? So that was the idea. And the goal were simple, basically to facilitate the transfer of those inventions to benefit society. As I said, I really think this is institutional responsibility. And then we need to recognize the fact that majority of our students are not going to have our jobs because we no longer retire and we don't die so quickly. So they don't have a job. So we need to educate them in other jobs that are available, really worthwhile jobs that are available, including those in industry. And finally, we recognize that the industry is in big trouble because their failure rate makes life too expensive. You can't make drugs if you are wrong 99 out of 100 times that you're doing that. And so perhaps if we take the academic culture that is try to do things that have not been done before, perhaps we can break this stalemate. So our goals was to move inventions that others have made at Stanford, to move them to pharma when feasible, or to clinical studies or clinical trial. And we really didn't care about value, so we didn't do market analysis or anything else. It had to be unmet clinical need, and importantly, it has to be something that we felt that we can do something about to increase its value. So if we felt that we didn't have any expertise around the table, let's say it would be a device or a software development. I don't have any expertise to do that, and nobody that I recruited to help with Spark did, and so it will be useless to take in. So the approach was to provide very little money. In fact, I'll tell you in a bit how much money we give. But the money is not given to the faculty. Actually, let me say, we provide $50,000 a year for a project. And it's a 2-year program if you make it. Now, those are labs that have $500,000 to $2 million in their budget. And they get it to their budget. They wrote a grant. And then sayonara, you know, they write reports on whatever they find. And as long as you publish and you do something good, nobody is telling them that there is a problem. Here, they do not get their money to their bank account. The money stayed in my office. And they get it towards milestones. So they say, I'm going to do so-and-so, it's going to cost me that and that. We agree on that. OK, here is the money to do it. And we don't pay FTEs. You have to put the skin in the game, too. So you pay your students, your postdocs, your fellows, whatever. And if you didn't get your milestone, the money will go to anybody else's project, because I have a budget a year, and I'm going to spend it. And so if you didn't make it, like in industry, you go down the drain. So the motivation is obvious. What's important, though, that the motivation for the faculty to actually show up for Spark is relatively little, because it's little money, and it's not theirs. So why do they come? And I think that the reason for that comes from what we have there. Is that, first of all, this education. For 2 years, we meet every Wednesday night, 5:30 to 7:30. That's why tomorrow morning I'm going to wake up at 3 o'clock California time to make it for the Spark program. And every other week, 4 or 5 of the projects are giving their quarterly report. So right now I have 34 projects in Spark. 34 projects that we need to keep in our heads. It's really a lot of work. Usually in industry, every 2 or 3 projects will be with one project manager, and here we have 34 between the 2 of us. They also have access to facilities that I mentioned briefly to Gordon about that there are certain facilities that are not typical for academic labs. You have actually, academic institution, you have some facilities here that are unique, including an amazing veterinary hospital, which is, if I have an animal, I made a transgenic mouse, and I want somebody to tell me what's wrong with this mouse, most of the times I will not know that there is something wrong with this mouse, because they are not spending that time in research, and they are not such experts in that, because we have one vet that is doing from horse all the way to, I don't know, monkey. And here you have that. And the other facilities have to do, for example, if you want to formulate a drug, or you are a basic researcher and you need an animal model for cancer, or you want to follow blood samples to look whether something is predicting of certain pathology. So having those core services is important, and that's what we provide access to. So I have 3-pronged responsibilities as a dean, and my second one relates to all these facilities. So we went from 10 facilities, service centers for Stanford, to 20 or to 19. And they are focused on a lot of things that help basic researchers do basic research, but also translational research. But the biggest thing that we have is mentors. There is no way that after doing one company for 1 year, I actually know anything about the industry. I just learned for a year what I don't know, and actually I just began to learn what I don't know. So the only way that you could do translational research is if you have people who actually know what they are talking about. So this is where we used to have Spark until last Wednesday, because we cited the second time by the fire marshal for having, in a room that had 96 people capacity, above 96 people. Spark started 7 years ago with 6 people. And we turned people at the door. Now we moved to a 200-room facility, and I said I'm not going to move to any bigger, because as it is, it's already lost one aspect that I wanted, which is everybody knows everybody by name. So now we have name tags, and people try to remember names, so when somebody is talking the second time, everybody kind of remember their name. This is one of our Sparkee, which is a faculty member of the department of, he's a nephrologist. And there are a bunch of advisors here sitting around the table, and then the people around the table are there. And actually, the photographers that didn't understand what I was trying to do told the people to move out of the way so it won't look like it's overcrowded. But it was, here are some of them. The advisors are coming from industry. They come every Wednesday night. So remember, one Wednesday night is a lecture, let's say on intellectual property, or how to get a company incorporated, or how to do high throughput screen. So they come to that lecture too. And it could be that the person who is coming to the lecture will be listening to medicinal chemistry, and he's a medicinal chemist. That's amazing. And the reason why they come, they tell me, is that it's fun. They find that these 2 hours that they spend on Wednesday night is fun for them. It's fun for them because they have discussions that they never have, because we academicians ask questions that were never asked, because we are naive, because we are not knowledgeable, but sometimes because we have an insight that they don't. They have a chance to talk with experts in their field about what happened in their companies that they were not able to share before. They have a chance to talk with smart people about things that they are expert in, so they have an opportunity to teach. And sometimes they have an opportunity to actually contribute. But the amazing thing, contribute in a way of intellectual property and actually advancing directly the projects. The amazing thing is that they come, even though they have a day job, they sign confidentiality agreement, and they are not paid, and if they invent, they sign an agreement that all the invention belongs to Stanford. That's very important. It's not that we are greedy, but if I was a third party, I was Pfizer, and there is an IP that is belonging to you and to an advisor from Genentech, there is no way I'm going to be able to negotiate a deal there, because I have to talk to Genentech and Stanford at the same time. Huge barrier, forget about it, not interested. And so they understood that. They went back to their company and got the companies to agree to it. So all that is a way to tell you, you can do it here, and you can say, well, you don't have as much industries that we do. It doesn't matter. First of all, you do have biotech around you, but also you have retired people who were alumni of your school, who came back to live here next to their grandchildren, or you have other people who are moving to this area because they like the weather. You just need to say that you want people that were in industries that are interested in doing it. And what happened to you, I bet, is what happened to us, which is now we have too many. So we started with 2 advisors, and now at the end of sessions when we have pitching for a project, we ask the non-advisors to leave the room, and I see around the table 30, 35 people who are advisors every Wednesday night. We have probably a roster of about 100 people who come on and off, but 35 in the room is very, very common. You can get it here, and I've been told before when I was talking about starting Spark elsewhere, oh, we can't do it because we don't have the experts that you do. We're not living in this area that you do. And my answer to that, if you want to make sure that you will never, ever, ever have biotech and pharma around you, then don't start Spark. But if you want to, then call these people who are having the expertise to come and help you. And then the natural thing is that if you start a company, you start close to home. And then you create the industry, and it will be kind of self-fulfilling. All right, I'm going to skip the rest. But the bottom line is, I don't have a chance to tell you about the projects that we have. But the bottom line is that we have finished working on 64 projects. So what is my milestones? That's what the dean asked. You asked me to give you money, and I actually asked for $1 million a year. He didn't hear the part about $1 million a year a project, which is what industry is putting $2 million a year a project. He didn't hear a project. So he gave me $1 million a year, and then it suddenly kind of over the years shrunk to $600 a year. So the rest of it I steal and beg from others, but I have very small budget. 34 projects. Every year, half of the projects are either licensed or moving to clinical studies or to clinical trial. And when they're licensed to pharma or to startups. So 10 companies or 11 companies started that are backed by VC, and 3 companies backed by angels. If you have 50% success rate, that means that you are actually failing. Why does it mean that you're failing? Because there is no way that half of the projects are worthy of going forward. It just means that you left many more on the shelf of Office of Technology license that you should have made an attempt to develop. So I wish I could grow the program further, but there is a limit to how much you can do with the resources that we have. But I bet you'll find the same problem, that your success rate will be very high. And the reason is that it's not that we are in academia so much smarter than industry. But we have the advantage, and that is that we have a job here, and our livelihood is not dependent on the success or failure of the last program that we worked on. I mean, gee, we're failing all the time, and nothing happens to us, right? We have a hypothesis is wrong, we write a paper about it, and we move forward. In fact, it's a success because we wrote about it. But if you failed in a program development, you know, your program is closed, the resources for that is ending, and you are out of job. So there is a perfect match of cultures in academia and industry, and there is a terrific opportunity to really do the right thing, which is to take these early discoveries that were funded by the public and then try to do something good with them. And it's really within reach. It's not, you know, so far from reach. So we started, we are happy to help institutions start their own Spark program, and I'm happy to give you more information. In fact, I'll give you the slides so you can look it up. It's not rocket science. It's actually easier than you think, and you don't need to know it. You just need to know that you don't know it and that you need experts to help you figure it out. And then you can do good stuff and fun stuff too. So thank you for your attention.